Okay, let's look at development pipeline. So we've looked at last week on this particular process from target identification to hit lead, lead optimization, preclinical trials on animal work, clinical trials, and finally drug approval. We've looked at this particular topic last week. Okay, and now as a pharmaceutical company, if you think about it, you need to spend a lot of time and money to actually trying to develop this particular product or a drug, so to say. Okay, so but why does companies still do it? Basically, what we've not covered last week is the concept of risk. Okay, so in the process of drug discovery, normally the risk is very, very low. Okay, so in this particular project, in this particular stage of the research, trying to identify a target ID and whatnot are very, very easy, and therefore it's a low risk. So say for example, as part of your assignments, what you're going to do is basically you already have a target, okay, and you just need to design your own drug, but there's no significant investment involved in it, and therefore the risk for you to be able to produce a drug is very, very low. Okay, and therefore in terms of the balance between the resources spent and the output that you want to get, and especially if you're thinking about trying to get an output to the market. Okay, because when thinking about drug development phase, you need to think about all the process at the same time, as well as thinking about what would the single each process be. Okay, so if you're thinking about that way, target identification is very, very low risk, because regardless of whether it's a failure or it's a success, the risk is not there. Okay, if it's a success, then it's very good, so you move forward to the next step. If it's failure, then that's fine. You don't spend a lot of time, you don't spend a lot of money, so you can just recycle and do the process again and again without spending too much money and time. So once you reach your target validation, now the risk becomes slightly more higher than trying to just find a target that can be useful for you. Okay, and then in this particular stage is more on the discovery stage. You're trying to find what would be the best component, what would be the hits, which structure actually has the potential to be used as a drug of a certain disease of your target. Okay, and once you manage to find a more specific target and then move to hit or lead identification, now the challenge is even higher. The risk of failure now is higher, because you have spent more time to try and create your own database, for example, trying to get everything working properly. Okay, and similarly, it goes to lead optimization. It's getting higher and higher. When it reaches preclinical stage, is still it's not the highest, but now it becomes quite high, because you have spent a lot of time and money in doing the basic research for thousands or even perhaps millions of components, because before you actually reaches this stage where you can just take the compound and do it on animal models. Okay, because now when you reach to animal stage, doing research on that is very, very expensive, and if you manage to pass that particular stage and move to clinical stage, it's even more expensive, and therefore the risk that you are taking, that the risk that the company is taking, is much higher. Okay, so say for example, if you looked at how COVID is, so there's a lot of how do I say this? There's a lot of process that was skipped because they didn't do a full, thorough investigation, detailed investigation on the animal work, and they did not do a full clinical study before actually getting the drug to the market. What happened was there was a very, very risky thing to do, but the risks outweigh the advantages that the vaccines may have if it were tested straight away to the population. Okay, so the risk was not really there during vaccine development, but nonetheless, for a normal drug development, the risk is high as higher you go to the market. So why does the risk is part of the consideration? Because if you think about it, in a clinical trial settings where the number of candidates, number of patients, number of samples are quite high, so it might be one simple drug, but you might test it in 20 or 30,000 humans. So even if like less than 1% of your samples or your test subjects produces or had a side effect which is very much significant, pretty much all the time spent is no longer useful, because the drug will definitely be blocked from getting into the market. Okay, so that's where the risk is. So you spend a lot of money, spend a lot of investment, but if there's any failure within the last stage of the process, the risk that you are getting is very, very high. Okay, so this is also what the image on the right hand side is talking about. So as you decrease the risk, okay, so the risk is very high during the very low during the initial project development. As it goes higher and higher, the risk is bigger. Okay, and once you go into the market, once you reach into the market, the risk become smaller again, because you have fulfilled all the hurdles. Okay, as well as increasing value of your research. So why is this is also be put here? Imagine that you are developing a drug. Okay, so what you are doing now for your assignment is basically just far away from getting to the market. You are doing something as part of your learning. The value of the product, whatever you want to create after this, has more value to you as the developer. Okay, so it's more on personal value, it's not a market value. So the product that you develop, if you were to try and sell it, nobody's gonna buy it. Okay, but as you pass into preclinical trials, there's a significant value. If you pass clinical trials, the value becomes bigger and bigger, and once it reaches market, the value becomes very, very huge. Okay, so what does it mean by this? If you want to sell your product, okay, at any stage of the drug development phase or process, so if you reaches a certain stage, you may be paid. You can sell the product, you can sell the research to pharmaceutical company for them to take the risk for you, because sometimes you don't have enough money to actually bring the whole project to clinical trials or even to the market, because you need a lot of investment. But as a researcher, you might not have that particular access to money. So sometimes you just want to sell it based on what work that you have done. So at Universiti Malaya, so one of the professors that I know, Prof. Nur Saada, has managed to actually sell her finding to a pharmaceutical company and has generated millions of ringgit for the university based on her research. Okay, so drugs again is very expensive. Normally for a big pharmaceutical company, their budgets is into the millions, and therefore if you were to compare to the initial stage that even I am doing at Universiti Malaya, it is very, very low. Okay, so that's why the value is low. It's more on personal value, but the value in terms of how it's going to be market is not there yet. As well as if you look at the investment that the company needs to spend to just get one or two products out of the market. So a lot of money needs to be spent, and therefore as it reaches the final stage of the drug development, the risk is either getting bigger or smaller after clinical trials. Okay, and of course 2 to 5 billion, even though it's per annum, it doesn't mean that this is for a single drug. So sometimes big pharmaceutical companies that spends this much of money has a few targets, a few different drugs that they are trying to put into a target. So the research itself is prolonged. It's not a simple quick 1 year project. So normally it's between 8 to 14 years from the discovery stage by the researcher up until it can be found in pharmacies. Okay, And therefore in this particular stage a highly specialized expertise is needed, and the individuals that is working are normally a focused work, instead of what I'm doing now is just I'm a lecturer as well as a researcher. So I cannot really specialize in doing research because I still do need to teach on the basic theoretical principle about knowledge, okay, about the topic itself. And of course, the risk orientation of sponsoring company is different than me as researcher, because the research that I'm doing, I receive grants, and therefore, even if the research is not, you know, if it even if the drug research is not until the market, it is still okay for me because I do not have any risk on not getting the product to the market. So the risk is generally bore by the government in a public university settings. But the trade-off might be like what I mentioned previously, that one of the professors managed to generate millions from the investment that she received from the government, okay, from the ministry. Without the investment, she will not be able to generate this particular income. So there's always a trade-off. It doesn't mean that every single investment that the government puts in will brought about a new product, a new research, a new drug. Sometimes what's new is instead the knowledge, either knowledge that such and such doesn't work, or knowledge that from this particular point, you need to move into A star to make it going better. Okay. But for big pharma, so normally they are very conservative in terms of the length, because of the consideration on the regulatory review process that they need to take into consideration before the drug can be actually marketed. Okay. And of course, to generally drug development, the process, the investment and everything is unpredictable. Risk of failure is very, very high. So normally only big pharma can actually manage to fund a full clinical research. Okay. So in general perspective, normally about 1 in 5,000 molecules from drug discovery can go into the market. Okay. But nowadays, because of the advancements of AI, maybe the ratio will become larger. Okay. Like I mentioned previously, in my own work, I produced 1,600 molecules, and trying to see if any one of the designs were able to do something physiologically as a drug. So if you consider the 1,600 molecules that I have, and 5,000 molecule in general that is being investigated in the world, it's illogical to say that I am doing like 25% or close to 25% of what the whole world is doing. Okay, so this is just a rough value, excluding the advancements of technology. Okay. So some natural discovery, for example, penicillin G from fungus, the cephalosporin from fungus again, erythromycin, fungus, taxol from plants, Zeco and steroids can be found in multiple organisms. Okay, as well as using biotechnology to develop drugs, for example, Intron. So these are all I'm not going to discuss about it specifically in very detail, but suffice for you, if you are interested, you can just Google and read about it a little bit. Okay, so interleukins and insulin is from natural body production. Like for example, in humans, we do have interleukins, even Intron and insulin, even antibiotic therapy. But what does it mean by this is based on the knowledge, let's say insulin. So based on the knowledge that we have on how insulin is function, to produce insulin synthetically, to synthesize it, is very, very costly. So what people do is basically using biotechnology. So use bacteria to try, not try because it's already a success. So use bacteria to grow insulin, to produce insulin, towards the other day where it can be packaged and used by humans. Okay. While thinking about the design in terms of a synthetic design, where human actually design the drug itself, there's multiples like claritine, You have Zocor, you have Lipitor, Captopril, you have Zantac, we have Valium. So this is one of the kind of like an opioids, and ibuprofen is also one of the common drugs. Okay, and Zantac for allergic. Okay, and previously the way where chemists develop these drugs is via what we call as combinatorial chemistry. So I'm going to go into a bit more detail after this. Okay, however, even though the principle is there, but so far that I've seen, there's no success in terms of drugs that is being marketed. Okay, so success in terms of hits and leads, there's a lot even in combinatorial chemistry, but into the market so far that I've seen, there's none. And if you're thinking about computational structure-based drug design, so we have HIV, okay, where scientists actually looked at what are the proteins that is important for HIV to survive. They've looked at it, they did an investigation and designed drugs that were able to inhibit HIV, and is being marketed as well. And COVID vaccine, using the principle of mRNA to produce a vaccine that actually works in terms of producing or helping humans, helping our body to produce antibody to combat COVID-19. Okay, so these are some of the examples, including the pictures of the drugs. You can see some drugs are very, very simple, like ibuprofen. It's very simplistic, while Zantac, for example, you can see it tends to be more complicated with different types of atoms, different types of charges over here. Okay, so this is all the knowledge that if you learn chemistry and you learn a little bit of biology and medicinal chemistry, you can actually put yourself in any part of this particular stage of drug development. Okay, and finally, once you have a candidate, these are all the pharmaceutical criteria that normally you must investigate before you can actually submit the drug for approval to be marketed. As you can see, there's aqueous solubility, investigation on lipophilicity, permeability, metabolic stability, oral bioavailability, which is quite important for drugs that is more friendly to users. Okay, and investigation into the states of the drug, whether it's a solid or liquid, whether it's in terms of vapor and whatnot, and a lot of criteria that you actually need to investigate. Okay, so this is again some of the biological properties. We do have the requirements for that particular terms, so affinity, potency, efficacy, and selectivity, and what are the investigation that needs to be done. Okay, all right. So if you think about what we've covered so far, if you are a pharmaceutical company, you spend a lot of money to investigate this and that, getting some hits, getting some leads, and finally you might be able to get a few drugs, which is showcasing a very, very high potency in preclinical settings in animals. Okay, but sometimes what happens is when you switch to when you move to the next stage, which is a trial on humans, sometimes what these pharmaceutical companies saw was that the drug doesn't work. Okay, but what does it mean? If a drug doesn't work as intended, you have spent a lot of money, would you just give up? Okay, so these are some of the things that the pharmaceutical company can do. So number one, instead of thinking about mode of delivery, okay, so you might design a drug for oral. So your investigation in mice and everything is for oral, or perhaps a more specific mode of delivery, either intramuscular injection or subcutaneous injection, or supplementary delivery, or whether it's an eye drop delivery. Okay, so there's multiple examples of mode of delivery of a drug. So if it doesn't work, what you can do, number one, is use alternative modes. So what do you mean by that? So these are the common modes of drug delivery. We have oral, for example, for Panadol. We have pulmonary, transdermal, ocular, which is eye. You have from nasal, from your nose, your vaginal or anal. Okay, you have injection, subcutaneous, interperitoneal, intermuscular, or you have your buccal or sublingual region where you can try and deliver this drug. So as part of the process of drug development, sometimes you've already thought about, I want to design this drug for oral delivery, for example, because oral delivery is very easy. You can just market the drug. You don't need any experts to try and deliver the drug. Like, for example, vaccine delivery, last time we need to have a trained personnel, either a trained doctors or a trained nurses, to actually helps to deliver the vaccine to patients or to the bulk population. But if you produce a drug as an oral, we can just sell it as a over-the-counter drug. The drug can just be simply consumed and it works. Okay, but sometimes when you design a certain way, it doesn't work in the clinical settings. It might work in animal because animal have a different physiology, different processes. But when you move into human, which is your final target, it might not work. So if it doesn't work in one mode of action, you don't just stop there, because as you gets into a critical stage, you have spent millions of dollars to try and investigate full characteristics of your drug and whatnot. So what you can do is you can just move around, either search one by one, or you can move into what you think best based on your goals. Okay, so say for example, from oral you can move into nasal instead of all the others is nasal, because nasal is still easier. Nasal has been nasal delivery has been done or useful for example asthma patients. So you know that the mechanism is there, it works, and there are already cases where drug was successfully delivered via nasal or oral what do you call it, pulmonary routes. Okay, so you might try that instead of vaginal, anal, or different types of injection, because nasal route can still be done by the patient itself. Okay, so you can cut costs in terms of the how to deliver the drug. Okay, so hopefully you understand what I'm talking about for this particular slide, so you can move from one process or delivery mode to another until you finally find the best process for your delivery. Okay, so this is again just an example. If it's oral, before the drug gets or having an efficacy or drug response, there's a lot of stages where the drug needs to pass through. So if it's an oral, it needs to first get into your stomach, from your stomach, your intestine, your gastrointestine, and then from there it needs to get into your blood, from the blood transfer into your targeted tissue that has your biological activity, and therefore you can see the efficacy. Okay, so if oral doesn't work, what you can do is you can opt for GI tract, for example doing the anal delivery, okay, where it goes into your gastrointestinal. If this also doesn't work, you can straight away go into blood. This is where you can just use needle injection, okay, into your bloodstream, or whether it's subcutaneous, interperitoneal, or intramuscular. So there's multiple modes. If it still again doesn't work, then you get into targeted tissue. This is a more specialized, where if you like to watch medical drama, for example House, I like to watch House previously, okay, House MD, where you can actually see sometimes when they are doing surgery, doctors put drugs, doctor put this and that directly into the target tissue or target organ to make sure that there's a direct interaction between the drugs and the process that you're trying to affect. Okay, so this is just an example on the first method, the first alternative method, which is a changing mode of delivery. Second one is to convert your drug into a different form. Okay, so again, this is related to number one a little bit in terms of the formulation that is developed for oral delivery. So if one develop a drug for oral delivery, the formulation is different than if the drug were to be delivered either by ocular or by anal delivery. Okay, so the process is different, therefore converting it into a different form might be worth it. Okay, so example over here is we are looking at L-DOPA drug. Okay, so L-DOPA is currently being used to delay Alzheimer in elderly patients. Okay, so this drug is typically, if you try to deliver L-DOPA directly to your bloodstream by injection, it will not pass through what we call as blood-brain barrier, BBB. Okay, so it's a special membrane in our brain, pretty much, that isolates our brain cells from any other cells from our body. Okay, so if you try to deliver L-DOPA within your own by just injection, it will not pass through the blood-brain barrier. It doesn't reach the target organ, which is the brain, and therefore it will not have any effects, okay, in terms of preventing Alzheimer development. So what was done was to develop a more inactive form, or what we call as a pro-drug. Okay, so it is an inactive form of a drug, meaning that if you take this drug, you just give it to anyone, it will not have any effect. Okay, so it's not active ever. But the good thing about a pro-drug is that it is able to pass through the physical barrier that exists. So, and this pro-drug is what we call as changing either the formulation, or you change the structure of the drug itself so that it becomes inactive, but it can become active under a certain condition. Okay, so say for example, in this particular case, it's a pro-drug, it's inactive. If you inject it in your body, it does nothing. But once it passes through blood-brain barrier, what will happen over at the target site is there exists a specialized enzyme in that particular region that you try to use, so that the enzyme will convert your pro-drug into the drug that is active. Okay, so this is an example. So L-DOPA or levodopa is actually the inactive form. So what you want is you want to convert L-DOPA into dopamine, okay, which is the required drug. It's a natural compound, but nonetheless, because it comes from L-DOPA, it's actually a drug. Okay, so this is the inactive form. Levodopa, once it reaches to the brain, it's converted into dopamine, which is the active drug inside the brain that helps to delay Alzheimer disease progression. Okay, so this is the example. You have L-DOPA just now. It passes through blood-brain barrier. It still is L-DOPA shape, and then there's a few enzymes inside the brain. So these are the two enzymes. For example, it can convert L-DOPA to something else, which are not useful for your brain. But at the same time, there's a third enzyme that can actually convert L-DOPA to become dopamine, and therefore having effect to your brain. Okay, so this is the concept of developing an alternative. So again, this is just an example. So read through on your own time to just getting familiar with how it's affecting the brain. And we're going to look at the third particular process, which is optimizing drug. Okay, so in this particular concept is where you do structure activity relationship studies. Okay, so and this is particularly important for you to be able to understand, because this is part of your assignment. So what SAR means is you study the correlation between the structural feature and the activity of the compound against a specific target. Okay, so if say your target is protein A, and protein A is function as to convert molecule A star to molecule B. Okay, so this is the function. This is function protein A, but molecule B is giving, say, pain. Just for the sake of argument, so B is causing pain, A is not causing pain, but once it's converted to item B, it will cause a pain, and protein A is the function. So what you can do is basically, if you start off with say for example a benzene, you just want to try and see if benzene can actually bind to protein A, and from there you can actually see, oh, it binds, but when you test it in the cell, it doesn't really do anything. So what you want is perhaps you want to modify to add in hydroxyl group on the benzene, and again test on protein A to see whether it has an effect or not. And then from there you might see, before this, if you have 100 nanomolar of material A, in the presence of protein A, it converts to 100 nanomolar of B. Okay, but once you mix your drug that you try to design with protein A, you now saw from 100 nanomolar, the final product that you get is, say, 90 nanomolar only after 1 hour. Okay, so now we can see there's a reduction there's a reduction in the production of compound B, and therefore the pain that you might encounter now is reduced by 10%. So from there you know that, okay, hydroxyl group is very important for benzene because it causes reduction in a process, which is what you want. Okay, so because you know that hydroxyl is good, so you just leave it, you just keep this as part of a structure. So the feature now, instead of just a simple basic feature of a benzene, the benzene now requires an hydroxyl group to become a better drug. Okay, so that's your first investigation, that's your first inference. And then perhaps you want to do, what happened if you add 2 hydroxyl? Will it improve the process? But once you did adding another hydroxyl, you saw that there is slight reduction. So before this 100 to 90, now 100 to becomes 95 nanomolar percent. So the reduction now is increased from 10% reduction to only 5% reduction. So you know that, okay, hydroxyl alone is good, but 2 hydroxyls next to one another might not be good. It is still functioning, but you know, to a certain limitation. So you know that hydroxyl, you know, based on your experiment, you know that hydroxyl next to one another, ortho to one another, is not good. So you might want to change. Okay, perhaps we'll just move it to a meta position now. So you still have 2 hydroxyl, but now you change the position, and then suddenly when testing this, you manage to find that, okay, now it becomes from 100 to 85 nanomolar. So you know that 2 hydroxyl is actually still good, but the hydroxyl must be in the meta position and not on the ortho position. So you might want to investigate, because now you have an idea. Okay, 2 hydroxyl is good. So what happens if you have a 3 hydroxyls or hydroxyls in a para position? So you now have another design where you move the hydroxyl into para position, and whole and behold, you suddenly get 83% nanomolar conversion. So you know, okay, it's actually para position is way better. So these are all the structural feature that to Saturday relates back to the activity of a compound against a specific target. So structure activity is that particular process that I mentioned, where you have your original ones, and then from there, what's the output? You move to a second one to investigate if you add in more feature, or if you change the feature, what would be the effect, the biological effect. Okay, so SAR is actually an investigation between 3 parameters: biology, which is the effect on the design itself; chemistry, because you are changing the molecule; and statistics, meaning that if you change this, what would be the probability that it will change something. Okay, so you need to think about all of these 3 categories simultaneously while designing your assignment, while designing your SAR. Okay, and in terms of what assignments that you are doing, the biology comes into play via the ligand and target protein interaction. This is the study that you're going to do using Autodoc Vina. Okay, computational output data is the output from your computational analysis, again from Vina. So this one is you design using Autodoc tools, Auto ADT, Autodoc tools, designing your ligand and investigating generally on the target, and then you use computational data to get an output, and then from this output you try and make sense on the biological concept, the interaction and whatnot. Then you go back to chemistry to do chemical structure modification before you move back into this particular process. Okay, so the SAR assignment that you're gonna do basically involve all of these three processes, and that's why I would like this particular assignment be done instead of alone, but rather in a group, so that hopefully you can think better, you can design better, you can try and make sense of what the output is better. Okay, and the assignment, this is basically not really an SAR per se, because SAR involves biology, specifically doing enzymes activities and whatnot, but rather we call it as QSAR, okay, qualitative structure activity relationship. So you're just looking at computational data and try to make sense of what the output is. Now, the output that you get from VINA is in terms of delta H values. Okay, so delta H values, if you recall your first year chemistry, the more negative the delta H, the higher the tendency of that particular event to happen. It's like a Gibbs free energy. Okay, so delta H has the biggest influence in Gibbs free energy, and Gibbs free energy is talking about probability. So the lower the delta H, the higher the probability. So this is what we're gonna substitute with. So instead of doing a biological assay to get an exact data, so I want you to use a delta H value and output from Autodoc VINA to try and make sense. So you know that delta H, the smaller the delta H, the higher the probability. What it means is the higher probability reflects on the higher probability of your design drug to be able to inhibit or to interact with your target. Okay, so that's the concept. So as long as you listen to this particular lecture, then perhaps your assignment will be easier. Okay, so this is what you're going to do for your assignment. Hopefully you can just go through this lecture again and try to make sense of what you're trying to achieve for your assignment. If you're a chemist and you're trying to figure out without using computational tool on SAR of a compound, so what you can do is you can produce a combinatorial chemistry. So this is what we call as combinatorial chemistry, where originally you have one molecule. So you have four molecules. You have the yellow, green, blue and red. Okay, so you have four monomers. How can you design a drug utilizing only these four compounds, but you want to create a library of a lot of compounds using these four molecules? Okay, so in this case, showcase on the right hand side, you start off with green. Okay, and then each green you divide by 3, so that the first green reacts with yellow, second green reacts with blue, third green reacts with red. Okay, so from there, the first stage of combinatorial chemistry, you already get 3 different compounds from out of these 4 different monomer. Okay, and then again from one this monomer, you react it with another yellow, another blue and another red. You get the product, and then you get now a list of compounds. So you started off with 4 initial say molecules or atoms, for example. Okay, but after just 1 step, 2 step, you already have, how many is this? 1 2 3 4 5 6 7 8 9. Okay, so you already have 9 different compounds. So this one is 3, this one is 9. If you move into the third step, you will have 20 different designs of molecule very easily. So this is combinatorial chemistry, and previously this is part of the ways by which scientists do to try and investigate about what drug is working. Okay, nowadays there's computational tools, so it's easier to do it in silico rather than doing it in the laboratory, because it's more costly. Okay, so the approach for this combinatorial chemistry is for you to be able to develop a library using this combinatorial chemistry. So this is the library. For example, after 2 steps, this is what you'll get. It is a stepwise chemistry where you do the process one by one. Like this one, this is the first step. You get 3 molecules. You follow to the second step. You will have 9 different designs or different molecules. and then when you reach to the third step, you have 27. Okay, and nowadays you can just simply skip and do computational chemistry to try and make sense of it. So these three monomers, where can you or these four initial molecules, where can you get the information from? Well, technically a lot of things you can find it in different journals. For example, if you are targeting dengue, just for the sake of argument, so you saw that a lot of people has done research on dengue. So one person is doing compound A, another person compound B, another group compound C, and then another one compound D. So as a student, what you can do is just simply take A, B, C, D, and then you combine it into one molecule and see how it goes. Okay, so this is an idea on suggestion on what you can do, or perhaps you can do like I mentioned previously, just take out like one very simplistic molecule, for example naphthalene in this case, and then you want to modify naphthalene to see if naphthalene is actually working well. Okay, but do remember, based on your group, if your group is amino acid, then your basic initial molecule must be an amino acid. If it's carbohydrate, then it's carbohydrate, and so on and so forth. Okay, so selection of the group is very important for me to be able to understand what are you trying to achieve, whether your steps that you plan are actually matching to the goals that I'm thinking about when designing the particular experiment. Okay, so again, SAR, hopefully you understand. And part of the SAR assignment, so this assignment, even though you can't see it now, is looking at energy levels. If you know what this is, it's basically showcasing energy levels. So maybe one modification you get higher energy level, another modification even higher, and then as you go and go and go, you get higher and higher energy level in comparison to the initial energy level of the molecule. But then your final molecule that you design suddenly has a lower energy level, which is quite good. Okay, so this is the concept that you're trying to create, and see and be able to produce a molecule that has the lowest energy levels, and so that in terms of the probability, it's working better than the rest. Okay, so approaches in real life, you develop a library, computational chemistry, do a parallel synthesis, for example. You can generate a lot of large array of molecules simultaneously. Again, this is what I've mentioned. And for example, using peptides, carbohydrates, every single biomolecule that you have covered in 2004, it can actually be applied to this particular experiment. Okay, so once you have your library, what do you do? Basically, in the normal process, once you have a library, what you do is you just go and test it biologically, to see which one of your molecule can actually have the best effect. Okay, so just now we've looked at 9 or perhaps 27 different molecules. So what you do in real life scenario is you're gonna have a plate where you can put in individual compounds in here. Okay, imagine that ours is 27, so we just fill in all 16 boxes. So all 16 boxes will have your molecule, and what you do is you try and use a biological assay to see which one of your molecule actually have an effect to the same protein or to the same target. Okay, so this is kind of like a real life data that you'll get. You will see something like this. Green normally, of course it depends on what kind of test you are doing, but this is a general perspective. Green is showcasing that it's actually working. Red or orangey is showcasing it doesn't really work, or maybe it's the other way around. Okay, depending on what type of assay. So from here you can perhaps say, okay, this one works, that one works, that one doesn't work, that one is working. So you can go back and see to your library, and you say that, okay, this one works, this one works, this one doesn't work, and this one also works. Okay, so now from this particular assay, you already identify hits that you can bring forward to investigate the leads. Okay, so step number 2, you do a stepwise chemical, or alternative number 2 is, number 1 is parallel synthesis or combinatorial chemistry. Number 2 is stepwise chemistry. for your assignments, whereby you can just create one molecule at a time. Okay, so this is what I mean by benzene, and then suddenly you want to design benzene with hydroxyl and so on and so forth. Okay, so I'm not gonna go into detail again because I already described it in the previous slides. So if you kind of like be confused on this particular slide because I did not explain a lot of things, you can just go back and see the materials where I was talking about benzene and hydroxyls. Okay, so finally what you'll get is you'll get something like this. You have a list of compounds, you have the arrangement of these particular compounds, and then you have your values. So Ki is inhibition concentration or constant, but instead of having a Ki value, what you have a predicted delta H values. Okay, so maybe compound 18, in your case delta H will be -1, but number 2 it becomes 0, number 3 it becomes 3, and then number 4, once you change to one major component, it becomes -2. So now you know TB2 is good, so you might want to change now on R1, and you get slightly improved 2.5. So you want to try the alternative. So H1 you left it as H, but you move the CF from position number 1 to position number 2, and then suddenly you see a double of activity, which is good. So you're gonna keep that one, or perhaps you want to try and investigate from this, because this one still is showing slight activity, so you know that position number 1 does have an effect in terms of the activity. So you change it from a transfer moiety into a more OCH2 moiety, a methyl group. Okay, and see perhaps it gets even better. Okay, so again, the concept is what you need to investigate. I cannot say for sure whether you will see the similar trend or not. I'm not gonna limit on how many number of design molecules that you want. Okay, suffice that I said a minimum of 10 different designs. So if you want to do 20 designs, be my guest. 30, be my guest. All is up to you. This is part of your learning. Make a minimum of 10 for you to be able to really understand and grasp the concept. The more samples that you are doing, the more understanding that you will get in terms of how SAR relates back to the assignments. Okay, so this is just an example on what I did for my own work as a researcher. So what I did was using a computational chemistry to investigate QSAR. Okay, and basically over here you can see is AC001 through 3 and 4, and you can see over here there are common amino acids. Okay, so that one and this one. Okay, so this is my investigation, looking at different how length and changing of the amino acid changes the binding energy. So this is what information that you'll get from Vina. Okay, so from here you can see if I were to just take the bare minimum, which is WCFYFC, so this is the shortest, so that's the core for everything. So the value is -6.1. Okay, so it is within the negative energy, is showcasing a probability is high of the for the interaction. So what we did was we investigate what if you modify on the left hand side. Okay, so on the N terminal, so you add in another tryptophan over there. You do see, oh, the value increases a little bit, well, maybe it's good. Okay, and then you investigate again, you add in a second amino acid from the basic structure on the N terminal. Now what you saw was that, oh, it doesn't change much. So therefore, even if you keeps on investigating or modifying on the N terminal, you might not see much improvement. Okay, so that's one way. And then what if you modify it on the right hand side? So we do have RH one by one in terms of the investigation, and we do see the number is getting more negative until this one reaches -7. Okay, so that's the principle of what you're going to do. So but what you will have, instead of having just 4 molecules, you have 5, 6, 7, 8, 9, 10 at minimum, with what is the modification and how does the modification affects the binding energy. Okay, so this is what the structure and the activity relationship is for your assignment. Okay, and this is just biological essay about this particular topic. Okay, so we are still within the topic of what if drug doesn't work. We've looked at step number 1, we just change the mode of delivery. So number 2, you play around with the formulation a little bit. Number 3, you are designing or changing the structure of the API, the active pharmaceutical ingredient. Number 4, if you want to reformulate the drug, hoping that you can still keep on the same delivery mode, but just better. Okay, so a good example of this is actually Panadol. So a long time ago, Panadol is just Panadol. It's a single drug. If you have a headache, you have any small pain, stomach pain and whatnot, you just take Panadol and it helps you in pain relief. But nowadays, if you go into supermarkets or any other store, you can see there's so many different types of Panadol. So which one can actually you take? Because all of the Panadol are still oral based Panadol. You still need to eat it. Okay, whether it's a liquid, whether it's a solid, whether it's a solid, it's a big one, it's a small one, a different shape and whatnot. So everything is still an oral. So the mode of delivery is still the same. The API, the structure is also still the same. It is still paracetamol if you read the labels. Okay, so how do you change it so that the drug becomes better? So this is you play around with drug formulation. Okay, so there's a term excipients, additives. Okay, so you can just Google what they are in a bit more detail if you want to try and understand. Okay, excipients may perform a variety of function role in pharmaceutical products. For example, majority cases excipients is limited to kind of like to boost a certain activity of the drug. Okay, so for example, if you look at sorry, not this one. If you look at this particular example over here, erythroparabine and benzocaine. Okay, so benzocaine is the API, the active pharmaceutical ingredient, and the excipient is ethylparabine. If you look at the structure, they are actually almost similar, right? Except for this particular amino have been changed to hydroxyl. So I'm not sure what exactly is the function of this. I know it's a pain relief medication, but I'm not sure how exactly having an excipient helps in boosting the activity. But nonetheless, it does happen, and it happens either via random investigation or via a real knowledge base behind it. Okay, and if you look at this graph on the right hand side, it's just basically talking about how Panadol, the activity of the Panadol differs. You can see active fast in green, in the fastest state, which is fasting state. You can see that the absorbance rate for active fast is very high. Okay, but if you took the paracetamol after you had your lunch, the absorbance is low. Okay, so you can see this. Why? Because normally when you have your lunch and whatnot, there's already a lot of salts in there, so it might actually kind of like inhibit some processes from your drug. Okay, and hopefully you can just imagine what the information is and relate it to your own use case. Okay, so excipients can also be for general purpose, either promote alternative pathway for the drug itself, perhaps each. So you are still producing Panadol, but adults versus children. So children is a bit more difficult for them to swallow a drug. So therefore, you change the formulation so that it fits better in terms of who are your target audience. Okay, so that's why there's Panadol soluble, there's Panadol in a liquid form and with sugar, so that kids likes it better. Okay, because Panadol soluble is a bit pahit lah, so to say. Okay, and even if sex is different, so drug for male and female can be different, for example, so you might want to change the formulation. So you can also enhance the efficacy of a drug, for example, looking at the bioavailability. Okay, so this is um, the information on bioavailability. You can see the viability for active fast is very very fast, within 25 minutes, in comparison to 45, 55, and after 2 hours for a normal Panadol. Okay, it could be because of fast disintegration of the drug itself, and therefore fast absorption, or it can be just simply because it's fast absorption and not really fast integration. Okay, so these are all the information, and huge information. I just want to compress it into very very small points. You may also increase the drug solubility or suspension stability once you formulate it. Okay, taxotere, which is a cancer drug, is normally formulated using ethanol plus some surfactant, okay, or polysorbate. So surfactant, for those who identify or notice the word surfactant, is basically soap. Okay, and you know soap kills bacteria, so it can kill you. But anyhow, it is still used in drugs. Okay, so of course after investigation and whatnot. And you can also use another technology, for example, liposomes that contains a lot of cholesterol to help drug being delivered into cells. Okay, ensuring drug safety is very important. And even if you do drug formulation, the API is still the same, but the drug itself in general have changed its form. So sodium chloride, for example, is used in Panadol Active Fast. It helps in adjusting the tonicity of your body so that it absorbs better, but nonetheless it is not as safe for those who have had high blood pressure, okay, as I mentioned in our first lecture last week. Okay, and also it can be as part of a preservative, or for example surfactant, to prevent bacterial growth, to make sure that your drug lasts longer. Okay, it can also be an addition as a metal chelator, or disodium, for example disodium acetate. Okay, so that it captures metal and reduces oxidation, and therefore the drug bioavailability is higher, because otherwise, you know, some metals promote catalysts promote oxidation. So if you add in a chelator, it kind of like helps in terms of protecting against oxidation process caused by metals. Okay, so excipients can also be added as a targeting. For example, if you use liposome or virus like particles as part of excipient, so it actually helps in developing a targeted delivery drug. Okay, or it can be some other compounds, such as adding a color so that it looks more interesting. You're adding stabilizers so that the formulation lasts longer, or you can add in pH adjuster because you don't want your drug to be too acidic or too basic for human consumption. Okay, so we're going to touch again a little bit on excipients when we reach to our vaccine lecture, but for now these are generally what excipients is and why they are used in drug development as in general. Okay, so this is just an example. You can just read through and try to understand how it is used in a real drug formulation. If you do have any other issues, you can just come by and talk to me, because the lecture is almost like 1 hour, so it's gonna take becomes too long for you. So this is again basic information. I might ask about it in the final exam. Suffice for you to be able to grasp the concept for this particular lecture, and how do you relate it to all the other information that we have covered so far. Okay, so in summary, what we've learned covers these two particular CLO. Recognize the basic principle involved in drug research. We looked at the processes. We've looked at how drug is being developed in general principle, and how to utilize SAR, and we described a few types of drug. As you can see previously, some mechanism of action, mostly on interaction between the drug and the target itself, and we've looked at the basic principle of structure activity relationship. Okay, so this is not next Thursday. So basically that's it. suffice for you to be able to master the knowledge, to understand the knowledge, and finally be able to apply it in your final exam and utilize the particular knowledge for your assignments as well. Okay, hopefully by trying to apply the knowledge in the assignments, I'm hoping that there will be more knowledge retention, and it will definitely help you. Hopefully it will definitely help you in your final exam. Okay, so that's it for me for today for this particular lecture. For assignment, I'll do another different lecture. I'll do another recording so that it's separate segment and you can just focus on the assignment after this. Okay, so that's all from me. Thank you.